now I'm going to take a moment to introduce Jordan Rose from the Atlanta Science Festival to say a few words about the Atlanta Science Festival and what he's doing. So you're on.
Hello, everybody. Welcome to the Atlanta Science Festival. And just like the characters in this play are all about wooing each other with science, we too at the Atlanta Science Festival are about wooing you with science. And thank you for being a part of our festival and being a part of our science community. We have events like this happening all throughout the rest of this week and into next week through March 27th. You can register and sign up for other exciting science learning and entertainment opportunities at atlantasciencefestival.org. But I want to give you just Jordan's top 3 favorite events. We have the City Science Quest, which is an app-based scavenger hunt that you can do at home. That's going to be really exciting. And then there are self-guided discovery walks. Coming up this weekend, we have the Sweet Auburn District and the East Side Beltline. And you go to those places and you pick up a map, a beautifully designed map that's going to give you a self-guided walking tour through that neighborhood, pointing out awesome points of interest, lifting up the science and technology of that neighborhood that maybe you didn't really realize was there before. So discover Atlanta in a new way through those discovery walks. And then my number 3 is the Atlanta 40, which is also a self-guided experience. It's a mini conservation journey. So it's all happens on the web, and you'll get little cameo videos and fun poems about 40 different species that are important to the culture here in Atlanta. And you'll be able to learn about them and do a small micro conservation act. And the idea is that if you do 40 small acts, it really adds up to really helping us change our behaviors and fostering a safe future for our children and our children's children here in Atlanta and the rest of the world. So just a word of thanks to Atlanta Science Festival sponsors, including Emory University, Georgia Tech, the Metro Atlanta Chamber, the Arthur Blank Foundation, and many, many others. So we are Science ATL, the organization, the nonprofit that produces Atlanta Science Festival. So if you like what you see here tonight in our partnership with Horizon Theater, then please join us throughout the rest of the year for other exciting science events, not just during the festival, but anytime throughout the year at scienceatl.org. So thanks so much. It's really an honor to partner with Horizon Theater on this production, and we're really happy to work with them now and into the future. So enjoy tonight's show, and we'll talk with you all at the end with a real scientist.
All right. Thank you, Jordan. I'm so glad that you were able to join us to talk about the Atlanta Science Festival. I have a favorite I want to mention too. We have a new staff member named Nicole Palmietto, who has a company called Found Stages outside of her work at Horizon, and she does all kinds of immersive theater. So she's doing a show for the Atlanta Science Festival called The Shift, which is part theater and part escape room. And I plan to do it this weekend, hopefully with my daughter. So hopefully somebody will join us and come watch The Shift. It's playing the next couple weekends. Okay, now I'm going to introduce our special guest respondent, Charles Ford. Charles, come on up to the screen here with me. Charles graduated from Davidson College with a BS in neuroscience in 2012, and he began a combined MD and PhD program at Emory in 2015 and is currently completing his PhD in neuroscience under the mentorship of Dr. Larry Young. His dissertation work focuses on oxytocin and the neurobiological basis of social bonding, behavior, and cognition. So you have a lot to say about this thing. And we had him in the fall as one of our guests, and he has been very generous in bringing other people from his lab who are our other speakers this time. And I love his lab because part of what they're involved with is empathy and the science of attraction. And his personal thing, I think, is jealousy, if I remember correctly. So you have something to say. So I'll let you talk now about what you do and what your life is like.
Sure. Yeah. Thanks for having me and other people from the lab. Yeah. So our lab studies oxytocin, which is a neurochemical which conveys information in the brain. You might have heard of it as the love hormone before. And we specifically study how what happens in the brain when to substantiate social bonds, particularly monogamy, what you might think of as love. I have two projects. One is on sort of leveraging oxytocin in a translational way to ramp up activity in the parts of the brain that process social information, with the idea that this could be used to increase the efficacy of therapies for autism. And then the other project is trying to create an animal model for jealousy, which we don't currently have. Jealousy is actually pretty grossly understudied in the biological sciences, but we're working on that as well. So yeah, thanks for having me, and I look forward to seeing the show again.
Yeah, so that's great, because this show there's plenty about jealousy in this show. So we're ready to see tonight. All right, hello everybody, welcome back. I am going to bring Charlie Ford up to the screen with me here. So come on forward, and we will be the two of us. Amy on my seat. There we go. Great. So this is Charlie again, and he is a PhD candidate in neuroscience at Emory, finishing up his final year before, of all things, going back to medical school, which is unbelievable. So you don't have enough school, just got to keep going. But you're living the life of the scientists and PhD students in this play. So I'm just curious, as you watch the play, you've seen it before, you were with us in the fall, we tell our audience, what resonated with you, what felt truthful, what science was totally off the mark, and the playwright took lots of liberties, and both about the science, but also about the world of being in a PhD with a lab and all that good stuff.
Yeah, I mean, I think it captured the essence of being a grad student pretty well. I don't know that anybody really uses computer labs anymore.
Yeah, right.
They'll have laptops.
That's back from the old days. I think it was written a while ago, so he probably has to update that next time he does that.
But obviously, in the COVID world, spontaneous social interactions are virtually non-existent.
Yeah, right now.
No, that's sort of, I think, dynamic is pretty accurate. The opportunity for sort of interdisciplinary collaborations are often the source of kind of new insights and I don't call them breakthroughs, but progress. Those sorts of relationships between like faculties and students, they do happen. They're not treated casually. I don't know. I've personally never been involved in one, but I imagine that it would, particularly now.
To really complicate your life up a little bit.
I think it would be treated a lot like a scandal.
Might have been not so much back in the day, but now it's a whole different world. Yeah, when you do a play that's written a few years ago, it's like, some things have changed. We couldn't quite. That wouldn't be taken lightly.
That in particular, I think, has changed dramatically over the last 5 years or so. But a few decades ago, that was actually quite commonplace. I think it's a little stickier if you're talking about undergraduates and grad students, or grad students and postdocs, or a faculty member who's not your advisor. There are layers of complexity, but definitely your advisor, sleeping with your advisor would be considered a no-no.
Not a good thing. Yeah. Wouldn't move you up in the world. So were there things about the science that you thought he captured pretty well, or things that was like, that's not really accurate, but it worked for the show?
Yeah. I mean, I think for the most part, it was really quite accurate. I don't run little Petri dish assays. I know people that do that. There's a lot of subtlety to it. But no, I mean, for the most part, I thought that was all accurate. There is kind of a rule, like one of the universal pieces of advice that everybody gives you when you're starting out is don't make a knockout mouse model. Like, don't do that as your PhD, because it will take years and years and years to make it work, and it might not even do what you want. And it's very expensive. I think it would cost considerably more than $10,000.
Really? What makes it so expensive?
I'm not really sure. It's, when I was starting grad school 5 years or so ago, things were different. Now we have CRISPR in
Yeah.
some really, it's become a lot easier to use that stuff.
Right.
So I have a friend in the lab who makes genetically, like, knockout rodents. They're not mice, but they're rodents. And he can do it with some, he's been working on one in our lab for 2 or 3 years now.
And what does he make?
Like he knocks out a gene. So when they're talking about they were talking about, this protein,
Yeah.
protein M, and the way you see what something does is you get rid of it and then
Right.
see what goes wrong. And my friend has been making in the Young Lab, making a, he made an oxytocin receptor knockout prairie vole, which are like these little rodents, but they're monogamous. So we can study these complex social bonds.
Yeah. Why don't you actually talk a little bit, because we didn't really tell them what your lab did yet or anything about what you work with these rodents. Everybody in the lab is working with these prairie voles, right?
Sure. Yeah. So we study oxytocin, this love hormone, and the neural basis of monogamy and other social bonds. And the way we do this is we use these rodents called prairie voles, and they're like a little bit larger than a mouse, and they're kind of cute looking, and they form these little monogamous bonds where the male and the female vole, they raise the offspring together. They will, the male will like be aggressive towards other females that come in. If you stress one member of the couple, the other one will console it, and actually its stress will increase as well. And it really mirrors sort of what we have with humans, in what we would call social monogamy, where, I mean, really there's virtually no scenario in which pure monogamy would be evolutionarily advantageous.
Right.
So it's, we call it social monogamy, where you'll only have one partner where you have a mortgage with, or build a nest with, and raise offspring with, and share resources with. But when, that vole is out, foraging for food, or you're at the grocery store or whatever, and you need another, eligible mate, there's an evolutionary advantage to spreading your genetic resources
Right.
in other, in other baskets, putting eggs in others' baskets.
So how did our humans get where we are? If in the world that's not advantageous to hold on to your one partner, how did we evolve to what we socially believe is socially acceptable?
Well, I don't know why our society works exactly the way it does, and there are definitely other human societies where we don't have a strict monogamous system. And there are many scenarios where a large degree of monogamy is advantageous. The general rules is that it's based on like resource availability. There is actually one species of baboons that will switch between a polygamous and monogamous mating structure depending on resource availability. So if you think you're in this like Garden of Eden scenario, and there's like, lots of potential mates and lots of food, and there's no predators, then it just makes sense that you would like meet somebody, reproduce, and then go on your way, because like the little kids could, they're not at risk, they'll have plenty of food. But if you're in this sort of, very resource sparse environment, and there are predators around, and there aren't a lot of mates, and then one day you bump into the first eligible mate you've seen in years, the best reproductive strategy would be to hunker down with them, make as many offspring as you can with this one individual, pool your resources to raise them and protect them. And we think that sort of, resource availability is kind of what pushes mating strategies towards monogamy or polygamy.
So in your lab now, so you work with the voles, and I guess talk about what your specific research. You have two different research things. What are you doing with them in your lab?
So nowadays, to do most biological research, there has to be some sort of translational implication, some way that somehow what you're doing might one day improve the lives of humans, usually in some sort of clinical setting. So one of our projects is to use a drug to, if we give you this drug, it will and then you talk to another person, it will increase the release of oxytocin throughout the brain. So rather than just giving you oxytocin, which there are problems with,
Huh.
we can ramp up your already existing oxytocin system in the brain. Normally, we think of oxytocin as like this neuromodulator, and normally it gets released during sex, during childbirth, during nursing, all these times that you are like in contact with this other human being and you're forming this very powerful, frankly, you know, from a game theory, selfish perspective, like irrational bond with this other person. And the idea is that we could give this drug to patients with autism before they go into therapy. So for autism, you have these social deficits, and there are behavioral therapies that can be quite effective, but you really need like 30 or 40 hours a week to make them effective, which is grossly impractical.
Right.
And one of the issues with therapy for autism is that the therapeutic information is coming from another person, a social source, a therapist. So if we could ramp up the social information processing areas of your brain by sort of, you know, turning on the oxytocin system right before therapy, then maybe more of that therapeutic information would get through, and it would be more efficient. You wouldn't need, you know, 30 hours to really see an effect.
So it's not really about permanently changing the brain for the oxytocin with the person with autism, that it's using it for specific periods. Is there a problem with making that like a permanent thing where their oxytocin is elevated?
Absolutely. And I think that's, Larry and I just wrote a paper about this, but I think that's one of the fundamental problems with medicine and really psychiatry the way it is today. Neuroscientists don't think about like the brain as this soup of chemicals, and it's like, oh, you have this chemical imbalance, so let me, you know, put my finger on the scale over here and I'll lower dopamine or I'll raise serotonin, and then we'll just be back in equilibrium and you go about your life. That's not the way the brain works. We think about the brain now, sort of the metaphor we would use is more like circuits. But anybody who's ever had any sort of addiction, you know, to coffee or alcohol or whatever, like understands the basic idea that you can't keep consuming the same amount of a substance every day and have the same effect. Like you build up tolerance.
Right.
You know, if it's 1 cup of coffee every morning, eventually that's not going to do it for you.
Right.
And it's the same thing with any drug, antidepressants or antipsychotics or oxytocin or anything we try to do to the brain. So I think the entire field of psychiatry needs to switch away from this idea of here's a prescription, take it chronically, there you go, which has been around since the 1950s. And to like, admittedly, that was a big step forward from like lobotomizing people.
Right.
But we need to move towards this more targeted, how can we change the way your brain is wired through, you know, use of specific drugs in conjunction with therapy, in conjunction with life changes. Because once you change how the brain is wired, that can actually last. And then those behavioral changes or those, you know, cognitive changes can endure.
And are you studying, or as someone studying what you can't, that's a human thing, you're studying the voles, but somebody studies what actually happens in the brain when with the psychiatric aspect, with the therapy, so you've got something that makes it easier for therapy to stick, but is someone studying like what that therapy actually does to the brain, you know, itself, like in
so it's
humans?
really hard to study what happens in a human brain. And that's really why psychiatry and neuroscience, you know, can lag behind other fields, particularly with human stuff.
Right.
You know, the best we can do is stick somebody in a brain scanner, people call an fMRI.
Yeah.
And it's really a weak measure of brain activity. You're actually looking at blood flow, and it doesn't have very high resolution. There are people who do that and will give oxytocin or administer some sort of therapy and look at what happens in the brain, and you can sort of loosely see how connections between regions are changing.
Right.
And associate that with, you know, in PTSD, we find that patients have lower activity in this region, or it appears that like connectivity between region A and region B is less than it should be or more than it should be.
Right.
I think if we start mining data like that, we can start to generate hypotheses and test them for, okay, if I give this drug to this person and combine it with this therapy, maybe I get more of a lasting change. That's what we're seeing with ketamine and depression, or psilocybin trials for depression or PTSD. It's these more acute, we're going to give you this really powerful drug and this powerful experience 1 or 2 times. And that has this lasting change so that you don't have to just be taking this pill every day that you eventually build up tolerance to, and we have to increase the dose for.
Yeah, I heard about that. That's so interesting. But they probably have no idea what makes it be a lasting change. That's you know, the magic of trying to figure that out, I guess.
It's unclear completely. Part of it is just that's the way the brain works, right? Like you get PTSD because you have this one really traumatic experience. A stimulus enters your brain, and it's such a powerful stimulus that it changes the way your brain is wired. And there's no reason to think that we can't again change how the brain is wired through some other powerful stimulus.
Right.
Sitting in an office with your therapist is not that powerful a stimulus. But if you take an hallucinogenic, which can really increase how powerful that experience is, then it might be. And some of these drugs also promote what we call plasticity, or the ability for your brain cells to rewire.
Now, in the play, a lot of people do have these kind of eureka moments that turn out to be false. What eureka moments have you had in your work that, you know, that made you really excited and want to run outside and scream?
None that didn't eventually turn out to be false as well. That tends to be usually the case. And then, you know, your advisor's job is to have the wise, experienced perspective to keep everything in check. So the second project that I work on with a couple other students in the lab is on modeling jealousy, which is really terribly understudied, and we think of it as this sort of complex human emotion. But in reality, like we think it's this complex human emotion that involves, you know, requires love and things like that. But we also describe our dogs as jealous and things like that.
Yeah, right. So,
and it only makes sense that if there is a limited resource out there, that you would have some sort of emotional motivation to corner that resource.
Yeah.
So we think we can model this in these little voles, and we've tried I don't know 5, 6 different iterations of an experiment to try to identify that, try to develop a paradigm where we can have a male vole watch its female partner interact with a stranger male, and get jealous. We're having a hard time. It's hard. It's really, I did not appreciate how hard it was to develop a new behavioral assay. But we're making slow progress. We're maybe zeroing in on a brain region that might be involved, and then, you know, if we can figure out a brain region that's involved and have a good measure of a good behavioral output measure for, you know, if the animal does X more than normal, that means we can interpret this as jealousy.
Got it. You're trying to say, what are the outward signs that I can tag to jealousy? Is that what you're trying to say? Yeah. Okay. Exactly. And what is the experiment you set up to test this, or the most recent version of it? I guess I know you've had various versions of it.
Well, it's not the most recent, but the one that has worked the best would be we put a male and a female together in a cage, and they hang out for like a week, and then they develop this monogamous bond. We inject the female with estrogen so that she becomes sexually receptive. And then on test day, we'll put the male on one side of a cage with a barrier that he can see through, has a little hole that he can stick his snout in and sniff and investigate the other chamber. The other chamber we put is female, and we look at how he interacts with this chamber. And then we add a stranger male to it, who is very intrigued by this strange female that's an estrus, and he runs around and chases her. Sometimes they mate, and we watch what the male does. And we've tried a couple different things. One is having sort of a control animal on the other side of the female's chamber. So there's a third chamber,
Right.
And we wanted to see, you know, if you have a male has this monogamous bond with the female versus a male that doesn't know the female, is their behavior different?
Yeah.
Turns out that there are issues with how good a control that is.
Mm-hmm.
We've also tried leaving the male in the center chamber and having his female interact with a stranger on one side, and a novel female interact with a stranger on the other, so he has a choice between which chamber to investigate. That we actually got the opposite of what we expected. He spent much more time with the investigating the stranger female's chamber. You know,
Because of what you expected, huh?
Not,
So for this research, so do you have a real world application of this research, or is this one that doesn't require that?
I mean, nothing requires a real world application unless you want funding for it.
No, well, there you go. That's the point, right?
So really, it is, you know, fortunate or unfortunate that the reality of scientific research is is very expensive and it requires money. And really what you do is dependent on what you can get funding for.
Yes, right.
And we're very fortunate in the Young Lab to have a lot of resources. Larry is a phenomenal scientist who has many, many grants, and he can support many students doing many things. And he has money that he can give us to do these little pilot experiments that are just kind of interesting. If it's a younger faculty member, like the guy from the play, that doesn't usually happen. They have, you know, one grant with a very finite amount of money. They'll have one grad student, and that one project they're doing really, really has to work. Yeah. And then you do have this situation where the PI is like, well, no, you have to use flies because we can't afford mice. That is a very common scenario, but the economics of it change, you know, as you get more funding.
Right. And he's been at it for a while, so he's developed funding over time. Is he the primary grant writer for? I know the science is so grant dependent, you know. But at least unlike the arts, the science gets us in big, giant chunks. The arts get it in tiny, tiny, tiny little chunks, and we got to write a zillion of them. But you get it in like big money. So
very often, yeah, it's a lot of money.
Yeah, well, it's good. That makes it a little easier to survive and keep going. When you say, I had a couple of questions related to the play. He talks about the idea finally came to him when I left it alone. Have you found that in your work ever, where you just walked away from it and something popped for you?
Yeah, I'm sure that's happened. That's a pretty common thing, or at least not necessarily like leaving it alone. You know, you're in Puerto Rico drinking peanut butter, and then you have this idea, but working with other people from outside the field. Sometimes, you know, you get your best ideas presenting the research, and somebody from a different field comes up and asks you questions, and you're like, oh, wow, I can't believe I never thought of that. You know, doing the experiment differently. We, in that jealousy experiment, we had a Zoom, we attended a Zoom meeting with some mouse researcher or rat researchers in Japan who had this helping task experiment, and they were using this cage contraption that involved opening a door to rescue a partner, and it just the details don't really matter, but it was like hearing this other person half a world away doing this other experiment. We're like, oh, we should use those cages.
That's what it would work.
Talking about designing a new, you know, the 7th iteration of the jealousy experiment, using some of the materials and techniques that this person was using in Japan for a totally different experiment.
Yeah. And there is a lot, I know they talk in the play also about whether the old science being more compartmentalized. You've got biologists over here, you've got your physicist over here, but I know in your lab, there's multiple different kinds of scientists, right? In your lab, at least from talking to your peers that I was, what other kind of scientists are working in your lab and on what?
Oh, I mean, Senna, who was on here, is really I think he's getting his PhD in neuroscience, but his skill set is really computer science based. You know, there's another guy in our lab who's he makes viruses. He makes.
I know he told me about the guy who makes viruses. I'm like, I want to know how you make a virus. Oh my gosh.
And that's a tool that we use in neuroscience. Larry's had anthropology people in his lab and psychology people, clinical people. Yeah, I mean, the future of science is totally interdisciplinary, especially neuroscience, because I mean, you draw so much from biology and from psychology, sociology, and anthropology. It's all really rooted in philosophy of mind. But where so much of, I mean, virtually every field, but especially science and medicine, I think too, is going in this machine learning, deep learning direction. Something like genetics or proteomics, like from the play, you just have so much data. I mean, just orders of magnitude beyond anything that we can comprehend. Like, you can't, if you put it in a spreadsheet, Excel would blow up.
Yeah.
You need these deep learning algorithms to find the connections. That I really think is more and more the future of what we're doing. I'm not sure how that's going to play out, but the usefulness of that is just, it's increasing exponentially.
Yeah. So the idea that they don't trust computers, it's people just doing their work in the lab that is mentioned in the play. That's really kind of old school now.
I mean, there's some truth to that. You don't know how it's going to work until you try it.
Yeah.
I think right now we're in this sort of like symbiotic relationship where like you really can get all this data from computers and they can suggest hypotheses, but then you need to turn around and test them in the lab. And that was kind of outlined in the play. Yeah. And I think that's a pretty accurate depiction of a healthy dynamic between the machine learning, computer science aspect of things. Because really, computer science is in every research field at this point.
Yeah, it has to be.
And then the more traditional techniques. But I mean, over the last 5 years, the balance of power has very much shifted towards the computer scientists relative to where it was. There are papers that come out that are just very sort of computer science heavy. And it's like, oh yeah, well, we gave it the data and it told us this and we trust it. And
that's not so much. Yeah, you got to go back and test it after you make an assumption.
Well, it still gets published and it still gets cited. And, I mean, there's a lot of research out there that is, I don't want to say it's not good, but it could be argued against, or critiqued pretty heavily. And that is a thing. That said, if you're not doing something with computer science at this point, you're behind. Yeah, you're definitely behind.
Yeah. I mean, Senna was describing him. He's for those that were not with him the other day, but we're going to post his thing just talking about videotaping these voles in their activity, and then he takes the video and transcribes it into a mathematical model, which then transmits to the computer and creates an image on the computer that maps the behaviors. I was just like, oh my goodness.
So that's a perfect example of, so we're trying to make this jealousy assay, and we're putting this vole in an environment where we think he should be jealous. And we're trying to figure out how to read that. And it could be, how much time he spends investigating that chamber. That's what we were using.
Right.
We can look at the video and we can have somebody, press start and stop and time just how much time they spend investigating.
Yeah.
But that's not necessarily the best way to measure jealousy. It's not necessarily what a vole is going to do. So what you can do with something like what Senna's trying to make is you can instead film the whole interaction and use a computer to map mathematically the animal's body and classify different behaviors that we could never do accurately, much less, it would probably miss a lot of them. Right. But it can create these clusters where it's like, oh, the vole spends this much time doing this behavior and this behavior. And it's not interpreting it as, oh, it's grooming, it's scratching, it's sniffing, it's chewing. It's just totally objectively analyzing every single moment of data and every aspect of it, every part of the animal's body, to generate an objective analysis of how the behavior changes. So we can feed it that information and right now we don't have that technology, but theoretically something like this project could see a breakthrough when Senna's, algorithms and machine learning project really becomes successful and applicable. So I can just say, hey, Senna, give me your program. I feed it my data, and it comes out and it says, oh, this is what changed.
Yeah.
And that gives us something we can look at. We can go back and redesign the experiment based on that information.
Yeah.
That's the hope. And I think that is the direction that this field and almost every other is going.
The other amazing thing that Sarah was telling me about was about the use of, this is about, let's see if I get this right, using the virus to edit the gene and then turn it on and off with a drug that activates the virus. I was like, what? I've heard of CRISPR, but I had not heard of this,
So CRISPR is the technique that we do to implement that.
Is that what that is? When she's talking about the virus, is that the same thing? Is that what she was telling me about, or is it something else?
CRISPR is one of the techniques that we use to implement this. So CRISPR has this phenomenal ability to edit DNA and change genes. And then the question is, what do you want to change it to say?
Yeah.
So CRISPR was used in the patient in China, the child in China, to make it immune to HIV.
Right.
That scientist did it really unethically, and I think he was sent to prison.
Yeah.
But the way they did that was targeting the receptor that HIV uses to enter the cells.
Mm-hmm.
And the CRISPR virus went in and caused a mutation in that gene that made that receptor protein no longer function. Mm-hmm. Without the receptor protein, HIV can't enter the cells, and you have, effective immunity.
Right.
CRISPR is just a protein that we got out of a bacterium or an algae or something.
So CRISPR is a protein. That's what CRISPR is.
Well, CRISPR is a technique. It stands for clustered. The acronym stands for. But it involves a genetic sequence that then uses a protein. So it really has two pieces. One is a RNA, actually just like our mRNA viruses, target that will identify the part of the genome that you want to edit. And then the other is a protein, which is, I think, a bacterial protein that will then recognize the RNA. So we talk about it as a guide RNA that then guides the protein to come in and cause the mutation in the DNA.
Got it.
But you're just combining all these different techniques. So how do we get the CRISPR technology, the protein and the gene, into a cell? We use a virus. Viruses are very good at entering into cells.
Got it.
And then how do we get, instead of causing a mutation, we cause CRISPR to insert, say or not even CRISPR. What the virus can insert is a foreign protein, so that now this cell will express, say, a fluorescent protein, so that all those cells glow green. Mm-hmm. Or maybe we can get random cells to express the oxytocin receptor, so all of a sudden those cells will start being affected by oxytocin. And then we can go even farther, and we can say we're going to put in a gene that will express this protein, but only if this drug is present in the animal.
Right.
So then we can, by injecting the virus, which might only affect a certain type of cell, we'll give it a protein. We'll give that cell the ability to express a protein only if a drug is present. And that lets us, turn on and off different proteins in specific cells with the addition or removal of drugs. It's just this huge tool set that has been developing for decades and allows us to manipulate biological systems with, specificity that was totally unheard of just a decade or two ago.
Yeah, she was explaining that if you inject this, you want to study a behavior, you can see the before behavior, inject the drug, have the gene turned on, or no, I'm saying that wrong, I know, but have it turned on, and then you see the new behavior, and then it turns off. So you can actually, in fairly close time see the before and after effects, is what I thought she was explaining to me. Does that sound accurate of what she said? That's amazing.
Yeah. You can do it super fast. One of the coolest, most immediate versions of that is something called optogenetics. And it's people are trying to use versions of this to treat epilepsy and things like that, where we use a protein found in algae, which is responsive to light. And your nerve cells fire as ions move in or out of them and change the electrical potential of the cell. And these little algae proteins are ion channels that are responsive to light. So you shine blue light on them, the ion channel opens, and it lets ions go in that will change the electrical potential of that cell. So if we use a virus to insert into a neuron the gene for this algae protein, then on the outside of this neuron, you'll get these little light sensitive ion channels. Normally there's, no light in the brain, but if we can find a way to get light into the brain, in an animal, we'll put like a fiber optic cable in. There's some other research lines going on where maybe we can use fluorescent proteins, like the stuff that comes from fireflies, where you generate light emitting chemical reactions. Then we could, in a very targeted manner, manipulate. So in an animal, we can just shine light in the brain and we can turn cells on and off and do incredible things by manipulating that sort of very specific pathways in the brain by turning a light switch on and off. We can cause animals to kill pups, or normally infanticidal animals to nurture pups. We can induce a monogamous pair bond. We can make animals eat or stop eating.
It's a very scary technology, though, too, at the same time. I mean, it's a technology that has a lot of danger, from its application going forward. I know that there's a lot of talk about that scientifically, about how do we control or not control this and its application in humans.
It's unfortunately just the nature of the brain, is that we're not this free will thing that can just is immutable to external influences and makes its own decision. All we have to go in is stimulate a very specific circuit in the brain, and you can control people like puppets, like in theory. I mean, that's the direction we're going. And it has a lot of promise for therapeutic applications, where if you think of schizophrenia or PTSD or some disorder as a dysfunction of a particular neural circuit, we can go in and sort of fix that circuit like you would fix a gear in a machine. But as the ability to manipulate these circuits increases, obviously, with any other technology, the potential for bad actors to do these things also increases.
Yeah.
I'm going to jump in and bring us a question that was asked by someone who's on the other side. And this will probably be our last question. And the question is from Sunrisa Reed, and it says, if the vole had negative jealous feelings when watching his mated vole with another male, couldn't it make sense for him to distract himself with a different female on the other side? Have you explored that as a sign of jealousy? Seems pretty human to me.
Yeah, that is definitely one interpretation of that data. So it's a really good idea. That's one of the issues that we have with this paradigm is, in science, you have to watch out for designing an experiment where you can interpret whatever result you get to support your hypothesis. Like the hypothesis has to be falsifiable. That's the big knock against Freud and all of his theories. Even though there might be some validity to them, he could take, whatever your behavior was and say, oh, well, that's because, you either have these feelings or you're suppressing these feelings.
Right.
Whatever the result was, it could be reinterpreted to support his hypothesis. So, in that choice experiment, if the vole spends more time investigating his female partner, then he's jealous. But if he spends more time investigating the stranger, then maybe he's also jealous, just trying to distract himself. So that is like, we looked at the data and we're like, yeah, well, that was a really dumb experiment to run because, there was no way for it to give us a good answer.
Yeah.
And those are the sort of things that you realize as you, it's really hard to design a novel experiment, but that's why I'm just a student. I don't have to get money yet.
Yeah, getting the money is the hard part, right? So what's next for you? So you've got another year left of this, and then you were telling me, but I'll tell others what's next for you. What do you think in your journey?
Oh, I don't know. I hopefully will finish up the PhD about a year from now, and then I'll go back to med school. And I've already done the first 2 years, so then it'll be just the last 2 years, which are clinical. And we'll see how that goes. So 3 years from now, I'll be totally out of school, hopefully. And then probably a psychiatry residency, a research-based one that has a lot of time dedicated for research. And then who knows, then that'll be 4 or 5 years, and we'll go from there.
And what led you into this particular field, this medical/neuroscience? What made you decide to go down that road?
I started off in college really interested in philosophy. And I, philosophy of mind-body problem, consciousness, free will, things like that. And I studied that for the first couple of years and figured out that like I always wanted to base my arguments in some sort of empirical data rather than some thought experiment. And I eventually decided I would rather be doing science instead of just talking about other people's science. And that sent me down the neuroscience route. One of the issues with just doing straight science is that if you go that route, you tend to spend your entire life just working on one protein in one animal. And it's very, the farther you get in science, the more narrow your focus gets.
Yeah.
So I thought I would do med school too, so that hopefully you could be involved in that sort of like magical transition from the research to the application.
Yeah.
And psychiatry, it's in many ways it's lagging behind other medical fields in terms of progress. The real reason for that is that the brain is locked up inside this vault, and we can't just biopsy it, and it's much harder to manipulate. But we're at this point now where we're seeing this dramatic evolution of neuroscience and behavioral neuroscience, and our ability to understand the brain and manipulate behavior through brain interventions. And it's only a matter of time before we figure out how to translate those things into humans. Whereas, psychiatry, like I said earlier, is really mired in this 1950s behavioral pharmacology model of things. We're just going to give you a pill and have you take it chronically. Behavioral neuroscience now thinks in terms of specific brain circuits controlling specific behaviors.
Yeah. And
right now we can't manipulate specific brain circuits in humans. But if we can figure out how to do that, that would totally revolutionize our mental health treatment across the board. And I think, that working on that sort of problem would be cool. So it's,
targeted therapy. When you have cancer, you want to get over having to destroy the body, and the effort to heal somebody. Why go at it in that kind of very
targeted therapy for cancer is a perfect analogy. We used to just, and to some extent we still do, just load you up with chemotherapy and stuff that kills lots of cells in your body.
Right.
But more and more we're figuring out, for breast cancer, or prostate cancer, we sequence it and we find out, do you have estrogen receptors or proestrogen receptors, and which receptors? And we use the specific genomic signature of your tumor, not everybody's tumor, but your tumor, to curate a cocktail of drugs that will be most effective and have the fewest side effects for you. We're not doing that in psychiatry yet. We're just checking boxes. Do you have these symptoms? If so, here's the drug. But we will eventually be able to make it more specific.
Yeah, that's the great opportunity. I mean, it's a great time to be in the sciences with all of the new tools that are available, for sure.
If you look at depression, you get diagnosed as having depression if you have 4 or more of a list of symptoms. But you could have increased appetite or decreased appetite. You could have increased sleeping or decreased sleeping. So you could really have two patients who get the same diagnosis and they have completely different symptoms, completely different behavioral manifestations of their illness. And there's some research going on where people are beginning to figure out, actually, this group of depressed patients has this abnormality in their brain activity, and this other group has a different abnormality. And right now our response is just give them all the same thing. Yes. And there's recent research coming out, and there's so much potential for us to start, instead of diagnosing things from purely a behavioral standpoint, saying, you know, let's figure out what's going on in your brain that's causing your problem. And then we can compare that, you know, through this massive data set we have from treating, you know, millions of patients, what therapies will work best for your specific issue.
And is there significantly progress being made in that, in the being able to map those parts of the brain that are the triggers for those different psychiatric symptoms that way?
There is some. It's really all using fMRI, but there are some cool labs. There's one in California. I'm forgetting the person's name, but they're using stuff from wearables. And yeah, so I mean, there's certain things like if your usage of pronouns shift to use more first-person pronouns than second-person pronouns, that can be very predictive of developing depression.
Really?
You can get heart rate abnormalities. It's like they were talking about using wearables to help diagnose COVID before you started showing symptoms.
Huh.
And it again comes from this machine learning and the sheer volume of data this stuff picks up on.
Yeah.
If you combine that with, I mean, how many times do you touch your cell phone on the day? You just have this huge volume of data, and we can't make sense of it. But if you feed it into a computer that can process these hundreds of millions of data points, you can come up with patterns that are very predictive. So by utilizing these brain scans, even if our brain scan technology is a little weak, leveraging that with all the huge amount of data we're picking up from these wearables and such, then you can start to make, you know, clinically relevant predictions about people and what's going on. And even if we don't understand the cause, if you feed the computer enough information, it can say, hey, if, you know, we have grouped this group of patients into one type, you know, depression type 1, and this other group is depression type 2, based on this huge amount of data we have. And we just know from tracking their progress with different treatments that group 1 responds much better to psychological therapy and maybe transcranial magnetic stimulation, whereas group 2 responds really better to this type of drug.
Yeah.
And group 3 responds to something else. We have all the data at this point. It's really just a question of putting it together. We can see clinically relevant patterns, and by following them, have much better patient outcomes.
Yeah. And there's research going on in that, in alcoholism too, I think, right? You know about that? That's I've heard.
I don't know about that specifically, but I mean, there's truly, if you've seen Westworld at all, the show, you know, kind of the premise of it is that if you're collecting this huge amount of data on people, your ability to predict their behavior and understand what's going on is huge. We see the same thing with these tech companies as they monitor our usage of cell phones, or, you know, what we're swiping through on social media. Facebook and Google can give you targeted ads that seem to know you better than you know yourself. And so we talk about these algorithms and how detrimental that is when, you know, Facebook is using it for profit. But we have that technology and that data exists. And if instead we applied it to analyzing human disease better, we could also develop therapies that are just as targeted as the ads you see on Google. The technology is there. We just have to apply it in the right place.
Yeah, figure out how to harness it. Yeah. Well, that is a great way to end our conversation. Thank you so much. This has just been a fantastic conversation. Everybody that's watching, will put it out on YouTube and tag it with things that will hopefully get people interested in neuroscience to come and listen to this wonderful talk. So it's really a pleasure to have you with us. And we look forward to having you come to the theater sometime when we're opening.
I can't wait until you're back open. Thanks so much for having me.
All right, take care. There is going to be, if you're still on, there is a survey in your chat. Please take a moment to fill that out for Atlanta Science Festival, and we thank Atlanta Science Festival. Y'all have a good night and enjoy some more science over the next couple of weeks.